Hello.
Hello, sir.
Hi, how are you?
I am fine, sir. How are you?
Okay. Yeah. Um, why don't we wait for a minute or two for other people to show up? There's Barshan. Hello, Barshan.
Hello.
All right. How are you?
Yeah, I'm good. I
Good. Yeah.
posted my blog for the week.
Oh, yeah. Yeah, I saw that. And your pull request.
Yeah, and my poll request as well, yes.
All right. All right. So let's go over Barshan's pull request. So we have our pull request here, and this is from yesterday. So this is the title is Add Epic Preprocessing in Data Scripts. It's number 55. And so basically added tools to preprocess Epic CSVs. This is from the Epic dataset into compressed MPZ datasets in a PyTorch PyG dataset loader. And then these are the files added: requirements.txt, scripts in preprocessing dataset.py, which does the transformation, and that supports a conversion to a distance with threshold model of getting the distances and edges of the network. Epic process preprocess.py. This is parsing local and global index builders, all that. Then there's epic dataset.py, which is the that loads the NPZ samples into torch geometric data. So he's taking it's actually like a set of files that are, I guess they're CSV files, and then they're turning them into NPZ files and then turning them or opening them up in PyTorch. So that's good. I like that. Thank you, Barshan, for that pull request. Okay, so this is Barshan's GSoC post for week 2. So this is just kind of going over the different weeks. So yeah, I think if we do like we did a couple summers ago, actually we did this last summer as well. We post an introductory post on the project, post like a midterm update, and post like a final post on the project on the Medium for the lab.
Oh, okay.
Yeah. So the rest of these are fine. It's just that I want to be able to cross post those.
No, like I went through some of the past GSoC scholars for DevoWorm. Like especially I found Mainak and Mayuk. Those two guys had this habit of posting every week in the blog.
Yeah.
So I thought if someone wants to track my GSoC weekly progress, this would be a very good. And it's under a tag also as DevoWorm in my blog. So there's like weekly post if I can just go through it for the summer.
Yeah. I think that's good. Well, so we've done this in different ways, but that's kind of the way I want to do it now. Yeah. So this is good. And so this is the week 2 post. So this just shows the local and global analyses. So the single cell is the local, the multi-cell is the global. So you're pulling up like different samples and then you're pooling them into these global samples. Okay. Every embryo in the data set has the exact same tensor dimensions. Unborn or dead cell features are set to 0, so this is the cells that you have to deal with when they, before they divide or after they divide, and having that in the same frame. We talked about this on Friday with the sort of the idealization of the lineage tree, where when it was drawn out, it was like, okay, here's what this cell becomes. But in the EPIC data set, they're tracking cells in real time, so you get these intermediate states that don't align. This is the dynamic masking required so that the graph neural network or spatial temporal network can accept the entire developmental tree of any embryo. So this is the graph here you showed on Friday, the dynamic masking heat map with the active cells over time. Okay. So then the local and global modes avoid any extra padding or dimensionality overhead. Okay. And then the point cloud, down to that. there is the point cloud that has these points, and there's a point cloud. And we talked about how we use point clouds in the group, or how we've traditionally used point clouds, and that's, you can grow a point cloud, you can isolate specific cells, or you can expand it to the entire embryo. Okay, and then of course now once you have the data in that form, the MPZ files and the sort of normalized positions, then you can import that to PyTorch geometric, and you can do things there. Yeah, and then
that's the enter point.
Yeah, so PyTorch geometric is mostly used to collate multiple graph samples into a single large disjoint graph structure. Yeah, so that's where you're getting your graph generation.
Yes.
Yeah. So yeah, and then the visualization of the graph is handled. Is that handled in PyTorch geometric, or is that a different? Well, you'd have to use something.
It might have some library, but I think Plotly will visualize it much better than PyG.
Yeah, there are other toolkits for networks, network visualizations.
Network there also.
Yeah. Yeah, so go down. Is that the end of the post, or is that?
Yeah, that's it. I have this tag on you.
Okay, so that's good. Thank you for that, Barshan. So why don't we start today with a few? We'll do a few papers today. I want to start with a theoretical paper and then move on to another paper. So this is from SciPost Physics Lecture Notes. This is from 2024. This was material for a summer school that is being held in France. This is by Bialek, who's a pretty top-notch theoretical biologist who does a lot of things with C. elegans. He's played around with mathematical models of positional information and other types of things. Very interesting work. But he considers in this paper, and this is kind of like the paper we did a couple weeks ago on the CompuCell 3D. This is called Ambitions for Theory in the Physics of Life. So this is where, he's a physical biologist interested in this physics of life idea, and then he's interested in building theory around that. And so, he has the abstract, and he goes through this, and there's, this outline here. So he's kind of framing this problem, reacting to complexity. Then there's optimal decoding, and then he talks a lot about matching distributions, which gets into this positional error idea. So this idea of positional information, and then the corresponding area of the positional error. And then network architectures. So the abstract reads, theoretical physicists have been fascinated by the phenomena of life for more than a century. As we engage with more realistic descriptions of living systems, however, things get complicated. After reviewing different reactions of this complexity, I explored the optimization of information flow as a potentially general theoretical principle. So it's really about information and information theory, more than really kind of thinking about the embryo as sort of a biological phenomenon, although you have to think about that as the way to generate that information. The primary example was a genetic network guiding development of the fly embryo, which is the work in Drosophila, which is another model organism that we use often, especially for this positional information idea. But each idea is also illustrated by examples from neural systems. In each case, optimization makes detailed, largely parameter-free predictions that connect quantitatively with experiment. So a lot of times in summer schools, in biological summer schools, they try to connect this with like some sort of experimental analysis or setting up and running experiments. So you'll often, get some lessons during the day or maybe during the morning, and then in the afternoon you go in the lab and you do some experimental work, and then you try to make that connection over the course of the session. So this is kind of why they do these lectures, so that people can then reflect on them as they do some sort of wet lab work. If we were doing a computational summer school, we would do some sort of a bunch of computational exercises with coding, but I would present this to you first to give you a frame for what you're going to be doing later. So, introduction, of course, optimal decoding, matching distributions, and network architecture. So, again, thinking about physics as a mode of inquiry is where we learn that qualitatively striking phenomena have correspondingly deep theoretical explanations. So when we see something in physics, we can describe it with equations, but we also have other things that we notice that we can't, we can describe in equations, but there's this overarching thing that's going on, and we want to build a theory that will explain those things. Because we can, predict things with equations, but those equations oftentimes sit in isolation, and we want to bridge that gap with theory. So in some cases, the relevant phenomena are quite mundane, and it takes time to appreciate just how surprised we should be. The everyday observation that the sky gets dark at night turns out to be one of these familiar but profound facts, as does the rigidity of solid objects. Today, however, the search for fundamentally new physics is concentrated in places very far from our immediate experience. One might be tempted to conclude that everyday phenomena are understood, at least in outline. So, for theoretical physicists, declaring something to be understood requires meeting a high standard. We expect a wide range of phenomena to be explained using a small set of general principles. Furthermore, we expect these principles to be summarized in compact mathematical form, and we expect this framework to be tested in quantitative experiments, often with little room for adjusting parameters, as we try to reach detailed numeric agreement between theory and experiment. By these standards, everyday phenomena of life are not very well understood. So, we want to have we have a high standard for understanding, much more than if we just kind of implicitly understand something to be true. And with biology, of course, we have experiments, we have a lot of experimental data, but very little theory, relatively speaking. And so this has been a problem in neuroscience, it's been a problem in biology, more or less, where we don't necessarily have all the theoretical tools that we would like. And so part of this is where we just don't have these principles worked out very well. In some areas, like in evolutionary biology, we have the principles worked out in much more detail than, say, like in development or in cell biology or molecular biology. But nevertheless, that's why we do this kind of work. It kind of gets into living systems and how it's different from sort of purely physical systems. And we want to be able to characterize life, but we don't want to anthropomorphize life, in doing so to get this greater understanding. So he gives an example here. He talks about this book, What is Life, which was Ernst Schrodinger's book from the 1930s. And he talked about, basically it's the first book that talks about biological physics. So this idea of life, which people still haven't really defined with any degree of satisfaction, with any degree of understanding necessarily. I mean, it's an open topic at conferences like Artificial Life or in the biological physics community. There are a lot of things you could include in life, a lot of things you could exclude from life, but sometimes you have these edge cases that make it very hard to have like a universal definition of life. And so what Schrodinger does in What is Life is just considers, these different biological systems that have physical principles, and then he tries to kind of bridge the gap there by suggesting how we might find a universal theory of life. And so since the time that book was published, we have all sorts of molecular biology data which give us a lot of very detailed information about the biology of different systems. But we still haven't really kind of found a satisfying explanation for what is life versus what is not life. And so, even thinking about single organisms and how they work is just as hard. And trying to figure out, what kinds of mechanisms exist in a biological system that would allow you to make a good overarching explanation. And so that's what he's getting at in here. Then he asks the simple question, how does the fly embryo make stripes? And so by the turn of this century, which is the 21st century, so around the year 2000, there was a clear outline for how this works. So again, we had this kind of worked out in the year 2000, so that's what he's drawing from here. To start, all the relevant molecules were identified, and this was one of the greatest triumphs using genetic methods to dissect a complex phenomena in living systems. So there was a lot of work done on Hough genes and Hough clusters, and that was used to describe this idea of different types of striping in the fly embryo that would lead to segmentation of the adult fly body. The molecules with stripe patterns of concentration are a group of 8 proteins that are encoded by the pair rule genes. Whether these proteins are synthesized is controlled largely by the concentrations of another set of 4 proteins that are encoded in the gap genes, so named because a mutation in one of those genes causes a large gap in the body plan. So these are concepts that come from way back in sort of maybe theoretical explorations even, but certainly experimental work. So this idea of the body plan actually goes back over 100 years prior. And this idea of striping and reaction to fusion goes all the way back to Turing about 50 years prior. So you have these different ideas that are kind of coming together in this work, exploring the mechanism and then bringing it all together. So the gap genes are so named because a mutation in one of these genes causes a large gap in the body plan. The gap genes are not only regulate the pair rule genes, but they also regulate one another, forming a network. So, we can use things like a body plan or reaction to fusion morphogenesis as a starting point. And we tend to find these sort of nuances and other types of things in different systems, and we can then explain them using sort of the same framework, but we kind of get more detail as we go along. So he's talking about this gap gene example where it forms a network, and then there are 3 inputs to the gap gene network that are provided by the mother when she makes the egg. The flow of information through the 3 layers of the molecular network is schematized in figure 2, which we'll get to. So this is figure 1, just shows this the striping in the Drosophila embryo. You see the striping here where you have you have the striping in the adult in different forms, and then you have in the embryo, you see the origins of it on the edge of the embryo and the stripes. And so this sort of looks like a rainbow along the edges of the embryo. It's actually encodes these different factors, Eve, RON, PRD, and then the nuclei are in gray, so it's differentiated from other cells, where these cells in these different colors are expressing these factors, and they're aligned in these stripes. So that's the idea, is that these stripes are formed by these coordinate systems in development and then become these stripes or these segments in the adult. Now figure 2 shows this process, but it's schematized on the left. So you have this connection between sort of these microscopy images on the right and the schema on the left. And so you start off with thinking about the earliest eggs and the maternal inputs. And so if you have a network explanation, if you schematize a network, it starts off with not having any sort of connectivity at all. And then, of course, you have your inputs to that network. So you have maternal inputs to the network, where you start to have these factors that kind of come into play. They can be provided or denied by the maternal source. And then once they're provided, then you start to get your connectivity. Then you have your gap genes, which form this coordinate system within the network. So it forms that connections in their polarity. And then the pair rule genes, which are sort of these separate nodes that then can be added to the network and turned on and off as needed. So this is kind of describing the growth of this network and the initialization of the network, but it also gives us this model for information flow. And so each node is a source of, or each node is a source of information, and their polarity is the source of information flow. So we can treat these networks as a dynamical system where information flows between nodes, and all we have to do is work out the flow of the information on these nodes, and then we end up with a description of this process where then we can manipulate the process. We can speed up or slow down the process, and we can end up with a corresponding phenotype. So this is maybe like something we do in the lab, right? We could introduce factors that would slow down or speed up the process. We could knock out certain genes, or we could knock out certain cells, and we could get some sort of result. And so that's kind of, there's been a lot of hand-wringing over connecting theoretical models with experiments, but at the end of the day, it really is just about abstracting away the most important things from the experimental model and then being able to predict and describe and maybe even control what we see in the biology. It's really not that hard. So this is kind of the theme of this paper, and so he gets a little bit into the information theory here and some of the dynamical equations of the network. So, he's talking about a genetic network. So these nodes are usually genes that are interacting, and they have to be turned on in a certain sequence, and they have to interact in a certain way to make this happen. So let's see, in a genetic network such as the one relevant for the fly embryo, drawing an arrow A to B, which is where you have different nodes labeled with letters. So A to B is this flow of information from node A to node B. Means that the rate of synthesis of B molecules depends on the concentration of A molecules. So you have a dependency that's formed by this information flow. So you have a differential equation that looks something like this, where Rmax is the maximum synthesis rate, and then your function A falls between 0 and 1, which is a normalized regulatory function, and then tau is a lifetime of B molecules, which is their rate of being in being in concentration. So we have to account for entropy, we have to account for the generation of new molecules. And so that gives us a number of things to work with at any one time. This is basically the idea, and he's talking about building out this network. In figure 3, he talks about how you have this relationship between A and B, and these are two concentrations that affect one another. So B is dependent on the concentrations and reactions in A that influences B, and so there's a flow of information from A to B. And so this is the graph here on the right, where you have A as the transcription factor concentration, which is the dependent variable, and then B, which is the target gene expression level, which is the independent variable. And as A increases, this transcription factor concentration at A, and transcription factors are produced here. It's usually some gene or some other thing that's being transcribed, producing mRNA, and it's building up at a certain level. So we have usually mRNA are transcribed, they're produced at a certain amount, and then sometimes they decay, sometimes they bind to other things, and so we have a certain pool that's available that is subtracting out that amount of decay or binding and adding in the transcription rate. So we can increase this concentration, or we can decrease this concentration from a baseline or a fold value, which is what we use experimentally, to get this variation in A. And then we have B, which is where we have this index, and so we go from 0 to 1. And so we can look at how this happens. So when there's a low concentration of A, it's usually due to a low expression level of B. And so when there's a low expression level of B, there's a certain effect. When there's a high expression level of B, that usually means the concentration of A is high. And so we have a couple of things we can look at here. We can look at the noise level. So the noise level happens when transcriptional noise occurs, and that's usually when there's some variation in the production of this mechanism. So transcriptional noise is actually quite important for, you know, in this process, because sometimes it adds some information. Sometimes it's necessary if we're dealing with environmental noise or other things that could otherwise, result in variation in the phenotype. So noise is important to characterize, not to mention that these processes have high tolerance, but they're not perfect. And so you get this noise level you have to account for. You also have cooperativity across genes, so sometimes A is not the only thing affecting B. You might have multiple variables affecting B, multiple nodes coming into B in the network, and you have to distinguish between the effects of A and the effect on B. There may be effects of, say, C or D or E all on B, and that's incidental to the concentration of A. So you might have a high concentration of A but a low concentration of C and D, and that affects the expression level of B such that this sigmoidal curve looks different. And so there's the sensitivity that occurs there. And so there are a whole bunch of things we need to think about with this simple relationship. It's not just about one thing affecting another thing in a network. Sometimes, especially if your networks are sufficiently complex, you have a bunch of things affecting one thing downstream, and then you have to disentangle those. This is why networks, they use the shorthand of the hairball, which is to say that when you look at a network plotted out, it looks like a hairball. All these connections are coming every which way, and it's the term hairball is a qualitative characteristic of the network, but the quantitative outcome of that is that there are a lot of interactions, and that those interactions introduce a lot of nonlinearity. So that any one node, if you sample its information content, is getting information from a lot of places in the network. And it's giving a lot of outputs of information. So you can't just simply use an additive model where you add up all the sources of information or split all of the information equally in the output edges that are going away from, like, say, any node that you're looking at. So this is, this just kind of underscores the complexity that we're dealing with, and that it's not just a simple statement to say that one thing affects another thing. So to give a quantitative description, we need to attach at least two numbers to every arrow of our schematic. So if we go back to our schematic, we need to attach numbers to these edges. We can't just leave them as statements of polarity and statements of interaction. So we need to add numbers because we need to be able to weight them. We need to be able to give them some sort of quantity, especially if we're dealing with information flows in a precise way. The first point made here is that something complicated could happen at places where multiple arrows converge. So if you have a bunch of things, a bunch of nodes that converge upon another node, so you have a node with a bunch of inputs from many different nodes. And we know that from, say, like the C. elegans connectome, where we have these rich club neurons, we have a lot of things coming into maybe like a rich club node or a rich club neuron from many places in the connectome. And so that could be, and we know that complicated things are happening at those rich club nodes because we know that those are kind of key in information processing for different functions. Now, because you're getting information from many different places, it's going to be heterogeneous. And because it's heterogeneous, it kind of suggests that that's a complex process going on there. Because when you have heterogeneous information, you have a lot of variation, you have a lot of different sources of conflicting information. So that's one point. The second point is that we have associated one molecular species with each gene, but there could be at least two, the protein and the corresponding messenger RNA. So again, we have maybe multiple molecular species that are working in concert or maybe in antagonistic fashion with that node. So, just because we have this nice, neat diagram, this schematic, doesn't mean that the process is nice and neat. Any one node can have a lot of things going on within it, and any one edge can have a lot of things going on within it. Number 3 is that we have assumed the dependence of synthesis rates on regulatory inputs, and that's instantaneous. But switching among regulatory states could introduce relevant timescales. So this is really where we have multiple timescales versus single timescales. So when we calculate like a synthesis rate or a decay rate or some sort of turnover rate, and we have these sort of accounting mechanisms for biochemistry, we kind of assume sort of equilibrium state. And this is true in physics. This is also true in physical biology and in biochemistry. We always assume the equilibrium state because it's easy to calculate, because a lot of times that's where we are in the space. And it gives us a way to deviate from that. So if we do an experiment, we often use a control where we have a single baseline, and then we usually use an experimental method to move away from that baseline. So we're always comparing the baseline with the non-baseline. And so that's why we have this. But of course, we may have nodes or processes that have multiple relevant timescales. So sometimes you have a slow process and you have a fast process, like where you have maybe synthesis, but you have decay and they happen at different rates. Or you have, maybe one process, which is the local process, and then a global process, which are the interactions. You might have some emergent process, which operates on a different time scale. In the embryo, there are many cells, so there's a separate copy of these dynamics at each of the 2 to the 14 sites. So again, every cell, technically has its own dynamics, although each cell usually works in a similar range of dynamics. At any one time, you can have very many different things going on. So we know that, for example, in a lot of experimental settings, sometimes people will fix the sample so that the cell division process is synchronized. So if I have a cell culture, for example, all my cells, I will try to synchronize their division so that every cell is in the same phase of cell division at the same time. So if I take a measurement, if I harvest those cells and I take a measurement of their mRNA, they're all at basically the same phase of cell division. So any effects of cell division on transcription is fixed. It's always going to be the same thing. We can just take out that variation and have a pure measurement. A lot of times, though, in actual organisms or in unsynchronized cultures, you'll have cells at various states of cell division. So when you take a bunch of cells and you measure their state, or you compare cells across the state, if you're doing single cell measurements, you'll get wildly different numbers for your model. And you could say, well, there's just a lot of variation, but actually that variation is explainable by what phase of cell division the cell is in. So it might be in a phase of cell division where it's reproducing a lot of copies of a certain gene or a locus, or it might be in a phase where it does very little of that. And that difference isn't just natural variation, it's variation in a process. Then during the relevant time period, there are multiple rounds of nuclear division, and the regulation of gene expression could interact with this cycle. So again, this is where we're dealing with cell division or nuclear division, and then we're dealing with that cycle as sort of a background process for morphogenesis. And so we have to kind of rule that out if we really want to understand how morphogenesis works. Even with these simplifications, a network of 4 gap genes with 3 maternal inputs could have up to 28 arrows, and in fact there is evidence that most of these exist. Certainly there is no reason to exclude any of them a priori from the theory of this network. But there are at least 56 parameters needed to describe the first few hours of development along one axis of the embryo of one particular organism. So if we go back to our image of Drosophila, this is a Drosophila embryo. This is one axis of the embryo, anterior to posterior. And along this axis, we have this process playing out. And the idea is that there are 56 parameters that are unique to Drosophila in its development, that are unique to this axis. So if we took the left-right axis of Drosophila, we're dealing with a different set of parameters, potentially. Not all of them are separate, but a lot of them are. If we're dealing with C. elegans versus Drosophila, there are other parameters that have to change because we have a different way that the cells divide, a different way that the cells polarize in the embryo, and so forth. And different genes that are controlling this, so different genetic networks, although they tend to be conserved across evolution. So you get very similar networks. Okay, so that's a little bit about this. Just a statement that there's when we're trying to build a theory of biology, we're dealing with a lot of parameters and we're dealing with complex dynamical systems. Okay, in the traditional core of theoretical physics, our models of the world have parameters, but somehow fitting these parameters do not seem to be the central focus. We assume that if there are too many parameters, there must be something that unifies them, and if we need to set these parameters precisely to non-generic values, we hope there are some extra dynamics that can make this happen more naturally. So that's kind of the idea behind that. I focus on parameters because we know from deep learning, say, that there are a lot of parameters in a deep learning model, billions of parameters. And it's kind of interesting to make that comparison between what we're doing in theoretical biology and computational biology and what people are doing in that community and dealing with this explosion of parameters. So I want to finish up by talking about how he thinks about complexity and how he thinks we should react to complexity. So in many ways, theoretical physicists' engagement with the phenomena of life can be classified by their reaction to this proliferation of parameters. At the risk of being cartoonish, let me list some of these broad classes. So this is kind of going through these different classes of how we react to this complexity, which is this proliferation of different parameters for different systems. So he starts with point 0, which is not necessarily serious, but it's worth thinking about, which is give up. Life is really that complicated. So a positive way of saying this is that living systems really must be described by models of many parameters. This is just a fact of life. And so the interesting theoretical problems concern how we infer these parameters from data, or how prediction may be possible even when parameters are undetermined, or underdetermined, I should say. So if we have a system where we underfit our data, basically, and we have, so basically we have a lot of natural parameters, but we don't have a lot of data. How do we predict? How do we do this at enterprise? And because, this is the sort of the rationale behind doing gobs of experiments, or just a lot of experiments. Because, if you talk to a biologist, they'll say, well, to understand anything, you need to do a lot of experiments, which is true, and this is why we do them. But we also need the model. The question is, when does the model come into play? When does it really become significant? And the answer is probably, with a lot of data. So for a long time, people didn't take theoretical biology or computational biology seriously because they didn't have the data they thought they needed to make these kinds of statements. But now, of course, we have a lot of data, and we have a lot of ways of getting data, and now it's less clear that that's really a barrier. So, it's an interesting question to think about. It's probably, it's probably not necessarily the case that we can't make any predictions, because we had these ideas that were kind of proposed 100 years ago or 50 years ago, and we're fleshing them out now with data. And maybe it's an exercise in just kind of taking old ideas and kind of dressing them up. But more likely it's about building building on a theory that was just kind of intuitive, but now we have the flesh to build on the bones. So that's good. Then he says here, it is not unreasonable to think that a theory of deep networks will circle back to influence how we think about the physics of life. This is a nice statement because it thinks about this, what the work that's been going on in the other direction, in the direction of theory about like artificial intelligence or about information processing in deep networks. And he's talking about deep neural networks and how we can learn from that. So again, with the parameters, how do we deal with systems of billions of parameters? Obviously, we have enough data to make those predictive. but we can do this with biology, so it's not too complex. Number one, we should study theories that remind us of the real thing and not try for quantitative comparison of theory with experiments on real living systems. So doing biologically inspired physics is not necessarily necessary. And so, as an example, much of the work on soft and active matter grew out of efforts to create simpler, better controlled examples of phenomena that we first encounter in the living world, from fluid membranes to flocks and swarms. And so a lot of that early work was biologically inspired physics, where you do physics that looks like something you'd find in biology and try to understand that without actually understanding the actual biology. And what he's saying here is we don't necessarily need to do that now. We have actual living systems to do it with. In the same spirit, neural networks are a source of statistical physics problems that are now quite independent of efforts to understand how real brains process information and learn from their experience. So this is kind of making the the parallel between biologically inspired physics and actual biological physics, and building neural networks in a way that helps us understand the brain. So, again, there's this parallel. And so we really kind of just need to engage with living systems head on, is what he's saying. Number two is the only real theory of how things are related to one another. So, sometimes people claim that biology is different from physics because biology is historical and physics is not. So biology is a historical discipline because we deal with common ancestry. We deal with this progression from development to adulthood. We deal with heredity. And ultimately, that's how it's organized. Physics is not organized like that. And so the idea is that, well, you can't apply theory to biology because it's historical and it's contingent on things that happened long ago, or it's contingent on things that happened in prior states that you can't observe. And physics, of course, is not that way. Of course, physics is kind of that way in some systems. But putting that aside, it suggests that we can't have a theory of life as we see it today. We could have a quantitative theory of the relationships among different life forms over time. Circa 2000, a number of physicists realized that population genetics and evolutionary dynamics could be seen as statistical physics problems, and this has been extraordinarily productive. So this is, actually a theme in a lot of statistical physics, is that, there is the statistical physics that treats everything as sort of this, stochastic system, or having this sort of independently distributed state. But there are also applications of statistical physics to these kind of historical systems and other types of systems that don't have the same assumptions as sort of where those methods were developed. So there are a whole class of statistical physics methods that people have used quite well to describe things from social systems to biological systems to technological systems. And here I'm actually talking about sort of the heart of network science. A lot of the measures we use to calculate centrality and find communities and determine other properties of these networks are all from statistical physics. Okay, so then number three is we are interested in relatively macroscopic behaviors, and these could be more universal than the microscopic mechanisms in the spirit of renormalization groups. So this is where we resample our microscopic data and we end up with these macroscopic states that may be more universal than things at the microscopic level. So thinking about things like emergence, where you see emergence in a lot of different systems, but the constituents behave very differently. They just return this sort of macroscopic behavior that is more universal. And so that's kind of one thing to think about, just simply because we kind of maybe make the assumption that if we see certain macroscopic behaviors that they have a similar microscopic underpinning. And that may or may not be true. The fact that living systems function often quite well can be promoted to a principle that selects parameters or behaviors circumventing details. So again, if we have like a certain explanation that we come up with, then we kind of maybe eventually ignore the nuance or the detail underlying that. And so this is something we want to avoid in biology because there are a lot of facts that maybe differ or between systems or a lot of conditional things that are going on. And, if we look at a visual system across different organisms or different types of environments or things like that, there are going to be differences in those details and in the parameters that control them. So here he talks about how bacteria navigate chemical gradients. Is it that they're counting every molecule that arrives at their surface, or are they using mechanisms that are more general? So these are things that we need to think about how to characterize these processes, how to kind of think about different behaviors, selecting different parametric regimes and things like that. So those are the points that he makes here, and so there are 5 different reactions to complexity, and I think those apply very broadly to not only to biological physics, but to sort of this idea of a complexity theory. All right, so that's all in that paper. I just wanted to go over that because I thought that was quite useful for thinking about networks and complex systems and dynamical systems and biology. And I think it's in the summer of the spirit of summer school that we do that this week. So it looks like Susan's here. Hello, Susan.
Hi.
Hi.
How many samples do you need to be biologically correct?
Well, that's a good question. Yeah. So if you're doing experiments in a summer school, sometimes they'll just use 3 to get the statistics to work. Like, if you do like
5 is better.
Yeah. But you can use 3 to get some get variance, right? And then you use 5, and then 7, and the idea is that if we want to look at something and say put numbers on it, we can do like as few as 3 replications or data points or whatever. Because you don't want to use 1 because you want to be able to reproduce something.
Yeah.
But then when you're dealing with like theory, how much data do you need for theory to develop theory, and then to confirm theory? So confirming theory, it might be more, but not a great amount. But then to create your own theory, you need a lot of data apparently, and that's something that you could debate with a biologist, with a physicist, and you might get different answers there. So
say I'm taking images of salamander eggs, the interior of them, and looking at blastocell development. How many eggs should I use?
I guess as many as you can get.
They lay hundreds of eggs.
Yeah, that's the thing. Like you can look at like 100, and the thing is like there's probably a lot of variation there. You could extract some general principles, but then you're going to always find exceptions. This is a thing with biology. You always find exceptions to the rule. Like in the 19th century, people proposed these laws of biology. Like you go back into the literature, you'll have like Kleber's law or other types of laws where they're largely biogeographical summaries or statements. And this was back when before we really had a really good accounting of say tropical biodiversity, where there's just a lot of stuff, a lot of data. So and so you can make these statements from limited observations, but you always run that risk of like having these exceptions to the rule, and that's always pernicious. I mean, it's like if you don't care about that, it's fine, but then is it a theory?
I'm just taking pictures or images of blastocell development.
Yeah,
that's all.
Yeah, it's not. It's sometimes, and for some systems, it's not that hard because sometimes there are things that are very like regular in biology, Now the underlying process is not, but you expect a certain outcome, and that's largely due to the complexity being buffered and other things. But yeah, I mean, I guess if you're developing a theory of what a blastocele should look like or what it is in one organism, that's you probably don't need that much data for that.
Yeah, well, it looks like this, like it's sort of an oval shape according to the literature, but there hasn't been that much done on axolotl because they're impossible to slice and dice. Like they're too yolky, the eggs, so you can't do microtome with them very easily. Dick came up with an idea of using microwave fixation for them that seems to work.
Yeah.
But with microtome too, you don't know if you've distorted it because you're slicing it.
Right.
So, yeah. So this is infrared light imaging. So, and it's the only thing that seems to work. So,
yeah.
So I'll hope that we can get this going. I don't know. The salamanders don't like to lay in June.
Oh.
Yeah. It's the wrong season.
Yeah. Temperature.
Well, it's also the light.
Oh, right.
It needs 12 dark, 12 light.
Okay.
Like even light dark cycle.
Yeah.
And that's not in the northern hemisphere in the summer. No, that's not what happens.
Yeah. All right. Well, thanks, Susan. Let me go to the other paper. We go, and then we'll end. We had a question. Okay, Barshan.
Audible?
Yeah.
Okay, so basically, it's not really related to the paper, but it's like academically, I had a doubt. So if I want to pursue a PhD or something in the future, seeing that I'm based in India, should I I have asked some of my seniors, and they say that it takes like 2 years to process everything, and if you want a PhD, you should start looking for it now.
Yeah.
So how should I start? Should I cold mail maybe a department, or is that how should I approach it?
Yeah, you should probably check out the departments and see what they're doing, and find one that does what you want to do, and then find out a little bit about what's going on there. You contact maybe some professors or some people who are doing that type of research and see what their sort of philosophy is, how they're if they need, sometimes they're not accepting students, sometimes they are, and try to work on that, work from that angle. And then you need to, when you write your application, you say, well, I've thought about this, and this is where I want to be, and this is why I want to be there, and these are people I might work with. So just kind of thinking about like where you think you might benefit from going and how you might benefit from that.
Okay. So does the PhD mostly depend on the professor, or is it like the department's thing?
It's usually the department, but also the professor. I mean, they have to agree to supervise you, but the department also plays a role in selecting people on their general strengths.
Okay.
Yeah.
Okay. Got it. Thank you.
Yeah, no problem. All right, so yeah, let's go on to the next paper. So this is actually a computational paper, and it's kind of different from the one we just talked about. This is on hyperagents. I think this is a nice paper because it kind of talks about some of the things in artificial intelligence that we kind of talk about in the Saturday meetings, but I thought it might be interesting to cover here because it does have some biological themes. So this is a number of people from across like Meta, different organizations, University of British Columbia, New York University. So it's Meta, Superintelligence Labs, et al. So hyperagents, let's go through the abstract. This is from the arXiv. So self-improving AI agent systems aim to reduce reliance on human engineering by learning to improve their own learning and problem-solving processes. Existing approaches to recursive self-improvement typically rely on fixed, handcrafted meta-level mechanisms, which fundamentally limit how fast such systems can improve. And so this is kind of a problem in this recursive self-improvement. We talked about this on Saturday, where we talked about some of the methods that the Sakana Lab in Japan is working on. So they're working on this idea of self-improvement in models. So the idea is that they observe their own behavior and they improve upon it, and they can do this. And we're kind of raising the parallels with like project management techniques like Agile and like Kaizen, which is an industrial application of, or it came before Agile, but it's very similar in the sense that it focuses on this recursive self-improvement. So it's doing some sort of self-reference and then it's improving itself. So this is something that a lot of times we want to put on board of an agent because we don't want to have human engineers try to do this, from a top-down perspective because it's slow, and we want to improve the adaptability of an agent. So this is something that they published on before called the Darwin-Gödel machine, or the DGM, which demonstrates that open-ended self-improvement is achievable in coding. So without going over this paper, this is sort of the source of their work here, is this Darwin-Gödel machine. I think Jeff Clune, one of the authors, was involved in that quite prominently. So starting from a single coding agent, the DGM repeatedly generates and evaluates self-modified variants, forming a growing archive of stepping stones for future improvement. So it's this sort of evolutionary process of evaluation of different variants for a problem solving a problem, or of a coding agent itself. So it's doing kind of what a genetic algorithm is doing. It evaluates those self-modified variants, and then it's producing or selecting the best one. And then, of course, this is used as sort of the template for future improvements. Because both evaluation and self-modification are coding tasks, gains in coding ability can translate into gains in self-improvement ability. So you can measure this self-improvement through coding improvement or coding ability improvement. So for an agent that's coding or generating code, the idea is that they would make different variants of themselves. They'd kind of select the ones that were best for the coding task, and then you'd measure coding improvement sort of as the quote-unquote fitness function of the system, although it's not a fitness function because they're not using this in the sort of canonical way that we use in genetic algorithms. However, this alignment does not generally hold beyond coding domains. Okay, so this is just an alignment problem that's being solved here. They can do this in coding domains, but it's a kind of a limited approach because that's an easy way to sort of do this, to characterize improvements in behavior, because it's something that's very easy to make quantitative, and it's easy for the agent to sort of self-evaluate. So in this case, what they're doing is introducing this idea of a hyperagent or hyperagents, which are self-referential agents that integrate a task agent, which solves a target task, and a meta agent, which is a separate agent, not to be confused with the company, but as a meta mechanism, that's an overarching mechanism, which modifies itself and the task agent into a single editable program. So you have this task agent, which is evaluating the task. It's sort of this local agent that says, if we're doing this coding task, it's going to just evaluate the coding task. And then the meta agent, which modifies itself, and the task agent, which is tasked with making this improvement or generating variation that can lead to an improvement. So they're two separate things that are kind of integrated in the same architecture. Crucially, the meta-level modification procedure is itself editable, enabling metacognitive self-modification, improving not only task-solving behavior, but also the mechanism that generates future improvements. So we can edit this. We can have this metacognitive self-modification from the meta agent, and that can help. It almost serves as kind of like the observer in a cybernetic context. So if we think about our system of improvement or self-improvement as a closed-loop feedback system, we then also have an observer at the top, which is this meta agent, which observes the feedback loop and will intervene if it can't find what it needs. So this is something we covered in our paper on the EGRT, which if you go back to our Saturday meetings, I covered at length. And basically our argument is that you can have these closed loop feedbacks that are sort of a form of self-improvement of a model with its world context. And then you have these observers that kind of supervise that process and improve it. So it's definitely a theme that kind of comes out of cybernetics. We instantiate this framework by extending GGMs to create DGM hyperagents or DGM-H. By allowing the improvement procedure to evolve, the DGM-H eliminates the assumption of domain-specific alignment between task performance and self-modification. So this is, again, where we have this assumption where task performance is sort of a proxy for self-modification. And we can assume that then if you're doing better on the task, that self-modification has been successful. And so this can potentially support self-accelerating processes on any computable task. So we're using the hyperagent as a way to sort of supervise this process to make sure we're not just assuming that any change in performance or any gain in performance is due to some self-improvement mechanism. We're actually supervising it in a way that's top-down as well. Across diverse domains, coding, paper review, robotics or word design, Olympiad level math solution grading, the GGM-H improves performance over time and outperforms baselines without self-improvement or open-ended exploration, as well as prior self-improving systems like DGM. We further show that the DGM-H improves the process by which it generates new agents. So this is persistent memory, performance tracking, and that these meta-level improvements transfer across domains and accumulate across runs. So this is, again, showing that this self-improvement is more pervasive than just simply a measure of performance on one isolated task. We discuss what safety entails in this setting and the broader implications of self-improving systems. DGM hyperagents offer a glimpse of open-ended AI systems that do not merely search for better solutions, but continually improve their search for how to improve. So that's kind of what they're talking about here, these hyperagents. And so they do hit on a couple of themes that are really interesting. Of course, they hit on this open-endedness theme. So open-endedness refers to the ability of a system to continually invent new, interesting, and increasingly complex artifacts, extending its own frontier of discovery without a fixed objective or a predefined end. And so open-endedness is a very important theme in artificial life, but also in complexity theory. Thinking about how to sort of drive forward innovation or changes without having an end goal in mind. And so if you think about it, it's very similar to what evolution does when it's producing biodiversity. It's producing things according to maybe some sort of fitness criterion, but there's no overarching goal about what life will look like. Life evolves as it evolves according to these local imperatives. It has common ancestry, but the benefit of that kind of approach is that it's able to explore new frontiers and sort of generate a lot of diversity in that direction to kind of solve a lot of problems that couldn't otherwise be solved if we had a single objective sort of driving the process. So open-endedness has been of great interest to people, thinking about ways to make this sort of exploration and improvement sort of I guess adaptive is the best word. So if you have a single objective and a single task, it sort of is like this performance to the metric or performance to the goal, and it becomes very artificial and brittle in terms of what the improvements are. When you have this kind of open-endedness, which is very similar to what you see in biology, it's very different. And so this is kind of a cool theme that they're kind of hitting on here. We don't talk about open-endedness, say, much in development, but we really do have open-endedness in the sense that we have sort of conserved processes. We have these processes that unfold and that are quite regular, but we also have sometimes we have things that need to be overcome in certain lineages, and so open-endedness is something that kind of interacts with evolution, and it'll be an interesting thing to kind of explore. Then there's self-improving AI, of course, going back to Hutter in 2003. There were these formal models of self-identifying agent or self-modifying agents where they developed. Schmidt-Huber, of course, was the first to develop the Gödel machine, which proposes agents that rewrite themselves when provably beneficial. And so this was something that was worked out theoretically, but not necessarily in a real-world setting. Of course, there was more work later on self-improvement through adaptive neural systems, in which agents modify their own weights or learning dynamics via meta-learning. And, there are other kinds of approaches like self-play to iteratively improve neural network agents, achieving superhuman performance in domains such as Go or chess. And which improved their play, but it was also one of these kind of hand-coded top-down approaches where you would have to define the algorithm and kind of improve it by human intervention. And so what they were trying to do in this work was to kind of make this relevant to the real world, but also make it so that it had not sort of a brute force intervening event, but you had this sort of observer of the system that was part of the system and that could respond in kind. So and then there's self-referential meta-learning, which is where systems that learn to improve the mechanisms by which learning occurs. So it's this sort of improvement on the learning process itself. Prior works have explored this idea in neural networks and evolutionary methods. More recently, several works have explored self-referential improvements using FM-based agents. The Darwin-Gödel machine and its successors instantiate recursive self-improvement through self-modification, primarily in coding domains. However, these approaches improve at improving primarily with encoding tasks only. So the Darwin-Gödel machine was just kind of restricted to coding domains where it's easy to sort of characterize the task and quantify things, quantify progress and quantify improvement in behavior. So that's kind of where they got how they improved upon that method, but then also the precursors of the Darwin-Gödel machine method. So then they just show the difference here between the Darwin-Gödel machine and the DGM with Hyperagents, which is basically where they take out the handcrafted instruction generation and the self-modify, which is self-modified within the program, and replace that with an observer that does this metacognitive self-modification. So that's all I have on that. Do we have any questions about that? Any comments? Okay, so well, thanks for attending. Yeah, so we will, if you have any questions about that later, we can talk about it offline. Susan?
Oh, I just a shout out to Dick, and I hope he's able to continue to write.
Yeah, yeah.
He's not feeling well.
No, he's not. All right. All right, well, thank you for attending. Hope Dick is doing, suitably. Morgan, did you have something to say?
Oh, no, I was just going to say, yeah, take care.
All right.
See you Wednesday.
All right, see you.
Cheers.
Thank you.
Bye.
Bye.